All right, BIM 100 students, let's get you ready for the final. We are going to do a super high yield review of Lecture 9. My goal is to get you totally dialed in on the concepts, and especially on how to crush those experimental data questions that you know are coming.
So let's be real. Staring at a gel shift or some chip data during a high-pressure exam can feel tricky. The whole point of this is to make that process feel like second nature. So when you see that data, you're not seeing confusion, you're seeing points for your final.
So here's our roadmap. We're going to start with a quick refresher on transcription factors, nail down the difference between activators and repressors, and then, and this is the important part, we're going to pivot to decoding experimental data. We'll wrap it all up by walking through a classic Pasquinelli problem step by step.
Right, so this first section is all about building that solid mental framework. You really have to get these core ideas down cold, because that's what's going to make interpreting the more complicated data not just possible, but easy.
Okay, first things first, let's do a super fast review on the absolute must-knows about transcription factors. I mean, these are the master regulators of the cell, so you've got to know how they work.
So, the one word in this definition I really want you to lock onto is modular. These proteins are not just some big uniform blob. No, they have distinct parts that do very different jobs. And understanding that modularity is the key to everything else we're going to talk about.
And here's that modularity in action. Every single transcription factor has these two critical parts. You've got the DNA binding domain, or DBD. Think of this as its GPS. It's what finds the exact specific address on the DNA. Then you have the activator or repressor domain. This is the business end. It answers what the factor does. Does it turn the gene on, or does it shut it off?
And another really crucial point, they almost never work alone. Most of these factors team up. They act as dimers. Sometimes it's two identical proteins making a homodimer, other times it's two different ones in a heterodimer. But that pairing is often totally essential for them to even bind to the DNA in the first place. Okay, so let's get into the two major jobs they have. Once a transcription factor actually binds to the DNA, what does it do?
The main difference here really just boils down to the state of the chromatin. Activators bring in help, these co-activators like HATs, that slap acetyl groups onto histones. This opens up the chromatin, we call that euchromatin, making it accessible for transcription. Repressors, they do the exact opposite. They recruit co-repressors, like HDACs, which rip those acetyl groups off, causing the chromatin to pack down super tight into heterochromatin, which slams the door shut on transcription.
All right, this is it. This is where the rubber meets the road for your final exam. Let's dig into the key experiments from this lecture and figure out precisely what each one is telling us.
This slide right here is basically your cheat sheet for the big three experiments. You have to know what question each one answers. So think of it like this. EMSA is your simple, in a test tube, yes or no question about binding. Does it stick? A reporter assay takes us inside the cell, in vivo, and tells us about function. Is this DNA sequence an on switch or an off switch? And CHIP, which is also in vivo, is all about location. Where in the whole giant genome is my protein actually sitting?
So let's start with CHIP. This is a really powerful in vivo technique. The best way to think about it is like you're taking a snapshot. You're freezing all the protein DNA interactions exactly as they're happening inside a living cell, right at that moment. This lets us see exactly where our specific protein was bound.
The way it works is pretty clever. First, you use a chemical, like formaldehyde, that basically acts like superglue, locking the proteins onto the DNA they're touching. Then you chop up all the DNA into smaller, more manageable pieces. And here's the magic step. You use a very specific antibody that will only grab your protein of interest, and you pull it right out of the mix. Everything else just gets washed away. Finally, you unglue the protein and analyze the DNA that was stuck to it. That tells you exactly which genes your protein was regulating.
Okay, let's apply this to a question you might see on the exam. The key is to break it down. We're looking for repression. So what does that actually mean in terms of data? It means two things have to be true. First, our protein, the progesterone receptor, has to be bound to the gene. We can see that as a band in the top row. Second, for it to be repressed, the gene has to be turned off, which means no histone acetylation. We see that as the absence of a band in the second row. So, which lanes meet both criteria? Well, if you look at drug 1 and drug 3, you see the receptor is bound, but the acetylation signal is gone. That's repression. That's your answer.
All right, let's switch gears to a much simpler technique. EMSA, also known as a gel shift. Unlike CHIP, this is all done in vitro, in a test tube. It has one job and one job only: to tell you if your purified protein will stick directly to a specific piece of DNA that you put in there with it.
And the idea behind it is really straightforward. A small piece of DNA on its own is nimble, it moves quickly through a gel. But if a big, clunky protein binds to it, that whole complex gets heavier and bigger, and it moves much slower. When you look at the gel, it looks like that DNA band has been shifted up to a higher spot. And that shift is your proof. That's the binding event.
Okay, now for the main event. Let's see how all these pieces come together in a classic, multi-part Pasquinelli-style problem. This is where she gives you data from a few different experiments, and your job is to put on your detective hat, piece together the clues, and tell one single coherent story. So, here's our mystery. The FOXP3 gene is turned on in thymus cells, but it's totally off in kidney cells. The big question is, why? To solve this puzzle, we're given 3 key pieces of evidence from 3 different experiments. Our job isn't just to look at each one, but to synthesize them all into a single answer. Okay, clue number 1, the reporter assay. This experiment tells us about the function of a specific piece of DNA. In this case, when we hook up this element X to a reporter gene, we get a ton of expression, but, and this is key, only in the thymus cells. In kidney cells, nothing. So what does that tell us? It tells us that element X is an activator site, a gas pedal, and whatever protein pushes that pedal is only found in the thymus.
On to clue number 2, DNA's footprinting. It's a simple idea. Wherever a protein is sitting on the DNA, it physically blocks an enzyme, DNase, from chewing up that spot. This leaves a protected gap in the data, which we call a footprint. And what do we see? We only get a footprint when we use protein extract from thymus cells. The kidney extract offers no protection at all. This confirms what we suspected. The acting proteins are physically there in the thymus, but they are missing from the kidney.
And finally, our third clue, the EMSA. This is the experiment that gives us our prime suspect. We test 2 different proteins, A and B, to see if they can bind directly to the FOX3 promoter. And look at that. We get a clear gel shift with protein A, but nothing with protein B. This points the finger directly at protein A as the factor that binds our promoter.
All right, it's time to put it all together. Let's recap the evidence. The reporter assay told us there's an activator element. The footprinting showed us a factor that's only present in the thymus. And the EMSA identified protein A as the direct binder. So, what's the final story?
And here is how you build that perfect, complete answer. You combine all 3 results into one conclusion. The reason FOX3P is expressed in the thymus is because thymus cells have a specific activating factor, protein A, that binds to a positive regulatory site, element X, in the promoter to turn the gene on. Kidney cells just don't make that specific activator, so the gene stays silent. That's the kind of synthesis that gets you full credit. And this is really the final takeaway. The goal isn't just to interpret the data you're given. It's to start thinking like a scientist. What's the next question? How could you prove, without a doubt, that it's protein A binding to element X? Maybe you could run an EMSA, but this time use a tiny DNA probe that only contains the element X sequence and see if protein A still causes a shift. Thinking about that next step, that's how you really show you've mastered this stuff. Good luck on the exam, you've got this.